In this video, we will study about salient achievements in crop biotechnology. Now, what is biotechnology? As the name is indicating, bio mean living, technology mean techniques. So, with the help of living beings and with the help of certain techniques, we can modify our crops. So, this is called as modification of the crops with the help of biotechnology. So, biotechnology plays a very important role in agriculture or in the farming sector. When we grow crop, we are interested in high yield, improving the nutritional quality, disease resistance, pest resistance, drought resistance, and improving the shelf life of the crops. Now, scientists, genetic engineer, researchers, biotechnologists, human beings, and all the farmers, they want to have high yield. Then, as human beings, we want to have food of good quality. Like if we see rice, wheat, and maize, they are our staple food. And if we eat them, they should be of good quality. And lot of pesticides and lot of chemicals are used to control the diseases and pests. So those chemicals, they become part of soil and they lead to soil pollution as well as water pollution. So they have to be checked. So there should be certain natural ways to control the diseases and pests. And some crops, they require a lot of water. So we want to create varieties which need less water, which are drought resistant. And when we transport the crops, like if we take example of tomato, if we transport and their outer wall get degraded after a certain time and we want to improve their shelf life, so this can be done with the help of biotechnology. So how biotechnology plays an important role in agriculture, we'll discuss. So crops which are modified with the help of technology, or we can say which has been genetically engineered with the foreign DNA, that crop is called as transgenic crop, or plants are called as transgenic plants. So we can say transfer of one or more genes into plants, and plants with foreign DNA is called as transgenic plant. Supposing we have this cell, it has its nucleus, and this is the DNA of the cell. If certain genes are added from outside like this here, right? Now this cell become transgenic cell because we have added a gene from outside, or we can say DNA segment from outside, and this crop will become transgenic crop. Now this gene which we have put, or DNA segment which we have put, that is gene of our interest. You can watch my videos of genetic engineering, cloning vectors. I will put the link in the description box. So there are certain techniques for transferring foreign DNA. One is we can transfer the DNA with the help of certain vectors. And most importantly, vector which is used for creating transgenic crop is Agrobacterium tumefaciens. This is a bacteria which is used for creating the transgenic crops. Or there are certain other methods like direct introduction, co-cultivation, electroporation, microinjection, and gene gun method. And this gene gun method is very popular method. Gene gun method, like with the help of a gun, a gene is transferred. So this is called as gene gun method. What are the various applications of transgenic plants? How we can create biotechnological crops? One is we can improve the photosynthesis. Now, what is photosynthesis? It is the synthesis of carbohydrates in eliminating green cells in presence of carbon dioxide and water, and that will lead to the formation of carbohydrates, and carbohydrates will improve the biomass of the plant, or we can say yield of the plant, or we can say fruit formation in the plant, and this will lead to more growth and good yield. So there is another process called as photorespiration. Photorespiration is respiration in presence of light, that is uptake of oxygen and release of carbon dioxide in eliminating green cells. So this process, photorespiration, will decrease photosynthesis. So naturally, if food is being synthesized or carbohydrates are being synthesized during photosynthesis, now there will be decline in the photosynthates, or we can say carbohydrates, or yield of the crop will be reduced. Yield will be reduced. So ultimately biomass will be reduced, fruit quality will be affected, growth of the crop will be affected. Now these two processes, they are controlled with the help of the same enzyme. This is called as rubisco. A full form of rubisco is ribulose 1,5-biphosphate carboxylase oxygenase. Now when carbon dioxide concentration is high, this part of the enzyme will be active. But when oxygenase is high, so this will be acting as ribulose 1,5-biphosphate oxygenase. Now ribulose 1,5-biphosphate carboxylase will help in photosynthesis, and ribulose 1,5-biphosphate oxygenase will help in photorespiration. So we want to reduce the photorespiration in the crops. That can be done by modifying this enzyme which is involved in the photosynthesis. So we will deactivate this part which is called as ribulose 1,5-biphosphate oxygenase, but we will activate this part which is called as ribulose 1,5-biphosphate carboxylase, and that will improve the photosynthesis. So ultimately improve the yield, improve the growth of the plant, and this has been done. We have transferred the genes from pea plant to tobacco for improving the rubisco genes. This will increase in photosynthesis, then this will lead to the high yield, and this is the tobacco crop in which yield has been increased. So if there are other various crops also where we can improve the rubisco activity by modifying the genes with the help of genetic engineering. Next role of biotechnology is providing resistance against biotic stresses. Now biotic stresses, biotic mean again living, stresses, certain stresses are there. Like when we are growing our crop, supposing we have a crop which is growing like this is a crop, any plant, this can be wheat, this can be rice, any plant. So this is the crop in which we are interested, right? But if certain other plants also grow here like this, which are not required. So when two crops they are growing, this is our interest crop, this is another crop. When two crops are growing, so they are sharing the same soil, water and air. So these plants will reduce the yield of this crop, right? And these plants which are unwanted crop growing between this main crop, they are called as herbaceous plant. Herbaceous plant, or we can call them as weeds, unwanted plant. We are not interested in these crops, but we are interested in this crop. So we want to kill these crops. One way is we will employ the labor, and labor will pick up or uproot these plants and save our main crop. But another way is we can use the herbicides. Herbicides are the chemicals like glyphosate, its common name is Roundup, then glucophosinate, ammonium, its common name is Liberty. But these are again chemicals, and they are going to pollute our soil and water, as well as they will affect the main crop also. So we create plant which are resistant to these herbicides. Like supposing this is the herbaceous plant and we want to kill with the help of herbicides, but our main crop should not be affected by the herbicide. So we want to create these main crops resistant to the herbicide. So we can say herbicide resistant in the transgenic plant. Transgenic plants are the plants which have the foreign genes or foreign DNA or transferred from some other source. So we can say herbicides are the chemicals, they are used to kill herbaceous plant. Herbicides, they affect photosynthesis as well as biosynthesis of essential amino acids. So there are two approaches. We can develop herbicide resistant plant. Supposing this is our main crop, again I'll make a plant just to make you understand. This is our main crop, right? And we are using herbicide to kill, we can say, this plant. We don't want this plant, we want this plant. So we are using herbicide. So this herbicide will not affect this plant, right? There are various ways. Like one is we can say target molecules should become insensitive to herbicide. Mean this plant should become insensitive to herbicide, or the target protein should be overproduced, or herbicide is affecting proteins in this plant, they should be overproduced so that other proteins they are not affected. Second is whenever this herbicide enters into this plant, this will not be toxic. So introduction of pathway for detoxification of the herbicide. If herbicide has entered into the plant, this will not be toxic in nature. This will not destroy the crop, because when we use the herbicide, our main crop is also affected, and we don't want that. We want only to uproot this unwanted or weed plant in which we are not interested. So we are using the herbicides or the chemicals. So we can modify our main crop. We can make this plant as herbicide resistant with the help of biotechnology. So there are certain herbicide tolerant transgenic crops which have been created by a genetic engineer or biotechnologist. So these transgenic crops, they are also called as genetically modified crops. GM crops mean genetically modified crops, mean their DNA has been modified. So example is like we can see here soybean. This is soybean. Now this is the maize plant, these sugar beets, then mustard plant, rape seeds, cotton, then rice plant, and this is the wheat plant. We can see here one by one. Now this is the soybean, maize, then sugar beet, rape or mustard plant, and cotton. This is rice, and this is the wheat plant. So various crops have been created as herbicide resistant crops. Now next application is insect resistant for transgenic crops or transgenic plants. Like we want to make plants naturally resistant to pests or insects. There are various pests and insects, like we can say there are bollworms, leaf borers, then shoot borers, then root borers, then we have weevils, many insects. They destroy our crops. So these are called as pests and insects. They will come and eat on the plant for their food, and they will destroy the leaves. When leaves they are destroyed, photosynthesis will be affected, and photosynthesis is affected, yield will be affected. Like root borers, they will naturally enter through the roots, and shoot borer, they will enter through the shoots. So their biotechnologists or genetic engineers, they have created certain genes for creating the plant resistant to the pests or insects. They have introduced certain genes for Bt toxins in these plants. Now Bt toxins, they are actually obtained from a bacteria. We will discuss in the next slide. So these pests and insects, they destroy our crops. Use of pesticide and insecticide to kill pests and insects for plant protection programs is used. Most of these pesticides and insecticides, because they are chemical in nature, so they will also affect our main crop in which we are interested. These chemicals will also contaminate the soil, leading to soil and water pollution. So to overcome these harmful effects, Bt toxin genes are introduced in the plants by genetic engineering. For this, a bacteria has been selected. This is called as Bacillus thuringiensis bacteria. This bacteria is rod shaped. Right? Now this bacteria has its own DNA. Then this undergoes sporulation by this reproduced by endospore formation. Right? So this will produce certain toxins called as Bt toxins. So this bacteria synthesizes a protein called as delta endotoxins, which are synthesized by sporulation, and these toxins, they are highly specific in their action, and these toxins can be used as safe insecticide. Its use is limited due to high production cost and due to its instability of crystal protein when exposed in the fields. Like we can see here, this is the bacteria. This is called as Bacillus thuringiensis bacteria. Now this is its main DNA, and this is the endospore being produced, and this is the Bt toxin crystal, right? So this is the naturally Bt toxin gene, we can say. So we can isolate from its DNA, like this is the DNA, we can isolate a piece of DNA from this bacteria, and that DNA, which is called as Bt toxin gene, and that gene we can insert in the crops. Now when we will insert this gene into our crops will become naturally resistant to the bollworms or pests or insects. And when bollworms, insects or pests, they come and eat our crop, now they will become lethargic or they will die, and they will not destroy our crop, and our, you can say, input of chemicals or insecticides or pesticides, which we are spending lot of money on these chemicals to control our crops so that our yield is proper, so that will be avoided and we can get benefits, or we can get high yield of the crops, right? So these because this is highly, you can say, its cost is high. So this also gets, you can say, its protein, they are not very stable, they are unstable when exposed in the fields, but otherwise lot of Bt crops have been created because this is very successful. Use of Bt toxin genes have been done. So first we have to take the bacteria, which is called as Bacillus thuringiensis. We will isolate its DNA. Now that DNA, we cannot always go and take the bacteria and isolate the gene. So gene which we have isolated, supposing we have isolated this gene, right? We have isolated this gene from the Bacillus thuringiensis bacteria. Now we can do its cloning. Cloning is production of identical copies, multiple and identical copies. So that can be done with the help of cloning vectors. For that you can watch my video of cloning vectors. So we can create multiple copies of this Bt toxin gene. Then this Bt toxin gene can be introduced into the host plant in which we want to introduce, and that can be done. Mostly it has been done with the help of Agrobacterium tumefaciens. So this actually bacteria has Ti plasmid. Again, I have shared a video of Agrobacterium tumefaciens vector mediated gene transfer. So this causes crown gall diseases in the plant. So Agrobacterium tumefaciens has been used for creating the transgenic crops. So we will introduce that gene into this bacteria, and this bacteria will introduce the gene into the host plant in which we are interested. Other method is by gene gun method, like gun shoots, and similarly that gun has, we can say, our interest of DNA, and that DNA is introduced into the target host plant. So we will get modified crops which have Bt toxin genes. Now these plants will naturally become resistant to pests and insects. So crops with Bt toxin, certain examples we have discussed. Now this is Bt papaya. Papaya is mainly destroyed by ring spot viral disease. So we can control, introduce in this gene, and this will be called as Bt papaya, genetically modified crop. Another is Bt brinjal. We know brinjal, this lot of pests destroy it, and we use lot of pesticides and insecticide to control the brinjal population. So Bt brinjal gene has been introduced, and this is destroyed by fruit and shoot borer. So we can control this borer with the help of this gene called as Bt toxin genes. Then cotton. Like this is the ball of the cotton. This ball is sucked by American bollworm, and when ball is not produced, cotton is not produced, then yield is less, right? So ultimately yield will be reduced, and we can control this gene, Bt toxin gene, and when introduced into the cotton, this is called as Bt cotton. And American bollworm, when it starts sucking on the ball, this will become lethargic, this will not be able to suck, and this ultimately dies, right? So we can control this pest. Then we have Bt tobacco. Tobacco plant is destroyed by bollworm and tobacco budworm. So we can again create Bt tobacco plant, and we can control naturally by introducing this gene. Another is tomato. We can increase the shelf life also, and we can create Bt tomato plants by introducing the Bt toxin genes. So this is how we are naturally creating the plants resistant to pests and insects by introducing the Bt toxin genes. So this is all about for this video. We will continue salient achievements of crop biotechnology in the second video also. If you have enjoyed my video, please like, share and subscribe. If you have any questions, please ask in the comment box. Thank you for watching.